Hello everyone. I welcome you all to the seventh lecture of the course Cellular Microenvironment from Oxygen Gradient to Gene Expression. In our previous lecture, we learned about transcription factors, which are among the most important regulators for gene expression. And we also discussed that even if DNA is accessible, a gene is expressed only when the appropriate transcription factor binds to the specific sequence of the DNA. And that transcription factor recruits RNA polymerase to the promoter region. We also saw that how if hypoxia initial factor functions as a transcription factor by binding to the hypoxia responsive element that is called as HRE and activates the gene involved in the cellular response to the hypoxia. However, another important question now arises. Once RNA polymerase reaches the promoter region, what happens next? Or how does the information stored in the DNA get copied into the messenger RNA that is called as the mRNA? So this remarkable process is called as the transcription, where the information is stored in the DNA that is getting transcribed into the messenger RNA with the help of RNA polymerase enzyme, and it represents the first step of gene expression. So in today's lecture, we will understand the complete mechanism of transcription, beginning with the RNA polymerase binding to the DNA, and ending with the synthesis of mRNA molecules using initiation, elongation, and termination step. As I have told you, the transcription is the first step of gene expression, where the information is stored in the DNA is copied into the mRNA. So why? I mean, So the learning objective of this lecture is like what exactly the transcription factor. This is a very fundamental concept. We should, in order to understand the whole transcription process, we should understand what is transcription factor. How does it recognizes one specific gene among nearly 20,000 protein coding genes present in the human genome? How does it finds the correct location on DNA? Then we'll also answer perhaps mostly important question is that how does HIF activates only hypoxia responsive gene instead of activating every gene inside the nucleus? And at that we will be at the end we will be able to answer these questions in today's lecture. So let's start with the transcription factor first. It's very important. So when we talk about the DNA, let's assume DNA is kind of library where lots of books are stored. So DNA is a library, then the books will be gene. So there is a large storage in the library of the book, right? So book is very a small unit for the library. And then RNA polymerase is kind of photocopy machine, right? And the transcription factor is kind of librarian. Okay, so the librarian will take book from the DNA from the library, will go to the photocopy machine, and then that will perform the photocopy of the book. So you can see that the RNA polymerase is doing something. That means that is transcribing the express gene in the DNA into the mRNA, and the transcription factor is the it's like someone who is doing this particular thing. So we'll understand these things here. Let's try to understand. So we can define a transcription factor is a DNA binding regulatory protein that controls gene transcription by interacting with a specific DNA sequence. So the transcription factor will decide where this has to bind to recruit the RNA polymerase. And this will bind only in that specific sequence of the DNA where gene is getting expressed. So if you see the gene DNA, so in DNA, if we focus on a specific sequence of the gene, then this gene will be having three sections like promoter region, where that is called as the upstream regulatory sequence. Then there is a transcription a start site from where the transcription will start, and then the gene expression or expression will get transcribed into mRNA. So this RNA polymerase, first what happens, this transcription factor will come to the promoter region and it will bind with this promoter region and it will recruit the RNA polymerase. Now RNA polymerase will, start the transcription from +1 site, and then the express gene which is there in the DNA that will get transcribed into the messenger RNA. So promoter is the DNA region where RNA polymerase binds and the transcription begin at the +1 site. As I have told you, this is something like that. Now coming to once how transcription factor recognizes the DNA, because the gene is getting expressed in the DNA. So who is telling or how this transcription factor is recognizing the DNA? So in our human genome, if you see, there are 3 billion base pair, right, in one sequence. So like there are two units in the DNA. One is called as the recognition motif, and another is called as the recognition mechanism. And this recognition of the DNA will happen with the help of these two. So the transcription factor basically, recognizes this recognition motif, and the reverse transcription happens with the help of recognition mechanism. So if you see here, the recognition motif is like you can see there are 6 base pair together. ACGTC or GCGTG, something like that. So what happens, this transcription factor first read this 6 unit of the base pair, and that is called as the recognition motif. And from where the transcription factor will bind, and then that will recruit the RNA polymerase. And in the recognition mechanism, it basically, works on that is a, I mean, these are the two transcription factor, recognition motifs and the recognition mechanism. So as I have told you that the transcription factor basically recognizes a short specific DNA sequence like 6 base pair or 6 to 20 base pair. But in recognition mechanism, what happens? The transcription factor have DNA binding domains that reads the shape and the chemical feature of the motif. So the working function of both of these are different. Here it is basically based on the shape and the chemical feature of the motif. Even though motifs are short, they are unique enough to allow a specific recognition. And this is how, based on these two recognition motif and recognition mechanism, transcription factor basically binds to the DNA. Now coming to the next part, that is activator and repressor. This is another important part. So activator basically enhances the transcription by recruiting a co-activator and the RNA polymerase. So activator basically, turn gene on. So it basically what happens, recruits the co-activator and then RNA polymerase to promote the gene expression, and it will transcribe this gene, and then the transcribed gene will be expressed in the mRNA, and then you will be getting high mRNA content. Then coming to the repressor, it is just opposite of the activator where it will turn the gene off, and it also recruits the co-repressor to turn gene off. And once gene is getting turned off, then the transcription will be off, and then you will be getting low or no mRNA. So these are two very important part of uh, transcription process. Now, how RNA polymers, how RNA polymerase enzyme is getting recruited. So here we will see that, let's say DNA was there, and then this transcription factor is getting binded with the specific sequence of the DNA at work from the promoter region, and this transcription factor basically recruits the RNA polymerase, and then this RNA polymerase will start transcribing the express gene into the messenger RNA. So the transcription factor act as a molecular bridge that recruits RNA polymerase, and the general transcription factor machinery to promote the promoter initiating replication. So if you see that, these are the steps that basically happens during the uh, uh, DNA to the protein, where chromatin gets opened. Already I've described all of these things in my previous lecture. Once chromatin will get open, then it is accessible for the promoter, and then the transcription factor, binding of the transcription factor becomes easy. Then this transcription factor recruits RNA polymerase, and then this RNA polymerase transcribe this expression into the mRNA, and then translation happens, and then mRNA is getting converted into protein. So we'll teach you these things in the later lecture, but here we have to understand till mRNA only. So why does the cell need transcription? That is very important question, and we are going to answer this question in this slide. So basically, if you see DNA, then the transcription happens with the help of RNA polymerase, and it forms the RNA. And then the translation happens, and then it forms the protein. That is a sequence of the amino acid. So transcription is a process by which the genetic information stored in the DNA is copied into the messenger RNA by the enzyme RNA polymerase. So what happens? This RNA polymerase enzyme recognizes the promoter region in the DNA that unwinds the DNA. That is very important. Till the time DNA is not getting unwinded, then it will be very difficult for transcription factor to get bind. And then that reaches the one strand of the DNA. I'll also teach these things which RNA strands is which DNA strand is getting read by RNA polymerase, and then finally it synthesizes the RNA. Okay, so how does this RNA polymerase read the DNA strands? It's very important. So if we see the DNA strand, there are two strands. One is called as the coding strand, that is called as the non-template strand. It goes from 5 prime to 3 prime. I will also teach these things in the next lecture or next to next lecture, why this is called as the 5 prime and 3 prime and or 3 prime 5 prime and all of these things. I'll teach you in the details. But right now, for the fundamental thing, just try to understand that there is a coding strand that is called as the non-template strand, which goes from 5 prime to 3 prime. And then there is a complementary strand that is called as a template strand, right? And that goes from 3 prime to 5 prime. So RNA polymerase always start reading the template strand. It does not read the coding strand, right? And as we all know that DNA is stored our genetic information, all the genetic informations are stored in the DNA. So cells will not, want to destroy the actual genetic information. It will try to preserve the genetic informations, right? So, but again, the gene expressed in the DNA has to get transcribed and then finally into mRNA, and then it has to become, translated to the protein. Then only because protein is the working functional molecules in our cells or in our body. So cells has to synthesize the protein, and in order to synthesize the protein, cells has to, transcribe this express gene, and that's why this transcription happens so that the actual genetic informations will be stored, and then let's say it is like a photocopied informations that will be there, that will get finally translated into the protein. So as we have seen that first the RNA polymerase reads the template strand, and like, let's say DNA coding strand that is called as the non-template strand, it is like this. So then the template strand will be complementary base pair of this. A will be T, then A will be binded with the T will be binded with the A, and so on and so forth, something like that. And as I have told you that RNA polymerase bind with the template strand, then the mRNA synthesis will, in mRNA, you will see that the base, the base which is getting added here is complementary to the template strand. So if it is T here, then it will be A. If it is A here, it will be U, because this is the mRNA. So in place of T, you will see uracil base, that is U. Okay. And this is how these things happen. Now, what is the direction of the transcription? So as we can see that the coding strand that is called as a non-template strand that goes from 5 prime to 3 prime, and the template strand that goes from 3 prime to 5 prime. So with respect to this, the synthesis of mRNA happens, and then you can see that the mRNA synthesis will happen again from 5 prime to 3 prime side. Okay. So you have to understand these things. And it's as simple as that. And finally, you can see this mRNA that you will be getting that goes from 5 prime end to the 3 prime end. Okay. And this is how the RNA polymerase reads the DNA strand. Now, coming to the most important part of this lecture, that is the structure of a nucleotide and the origin of DNA directionality. Why DNA is going from 5 prime to 3 prime, one end is going from 5 prime to 3 prime, and another is going from 3 prime to 5 prime. So if you see the, one nucleotide of the DNA, so it is having a three group, phosphate group, then there is a nitrogenous base that is ATGC, and then there is a sugar. So these three groups are there. Why this is why this one end is called as the 5 prime end. So it's very important. So if you see this sugar, in organic chemistry, we know the nomenclature. So it will start from 1 dash, 2 dash, 3 dash, 4 dash, 5 dash, because for this carbon, the base is added and it will get the highest priority. That's why this carbon will be called as the 1 dash. And we cannot go in this direction because this is oxygen. So we have to move towards the carbon side only. So that's why this will be, you have to nomenclate these things in this way. And that's why you can see that this phosphate group has been added at the 5 prime carbon. That's why this end will be called as the 5 prime end. And then the alcoholic group, OH group, that is called a hydroxyl group, that is added at the 3 prime end, and that's why this is called as the 3 prime end. So if phosphate group is there in one side, and that is added at the 5 prime carbon, that's why that is called as the 5 prime end. And we have seen that if this is a free hydroxyl group that is added at the 3 prime carbon, that's why this is called as the 3 prime end. And this is how you can see uh, first strand is this one. So ideally this image should be flipped in that way. So here you can see that the free phosphate group is here in the left side. That's why it is called as a 5 prime end. And on the right side, 3 dash end is there. That's why it is called as a 3 prime end. And this is how DNA strands look like. So this is your non-template strand that goes from 5 prime to 3 prime. And this is called as the template strand that goes from 3 prime to 5 prime. So how are nucleotides connected? So when we talk about the nucleotides, two nucleotides, so as we have seen that this phosphate group is not reactive group. Hydroxyl group is reactive group. Is that clear? So highly reactive group. So this hydroxyl group here, and then one oxygen of this phosphate group reacts, and there is a condensation reaction happens, and water molecules get released, and this forms a phosphodiester bond. And as we can see that the reaction is happening from hydroxyl group to the phosphate group side. Sorry, this phosphate group is also reactive, but the reaction will happen in that way. And that's why you can see that the phosphodiester bond is happening or getting formed from 3 prime to the 5 prime side, something like that. And this is how two nucleotides are connected with the help of phosphodiester bond after releasing of one water molecules. So this creates the phosphodiester bond and giving the DNA strand as a natural direction. And similarly, you can see why do we write DNA as a 5 prime to 3 prime? That is template strand, because this 5 prime end is called as the free phosphate group. And that's why this is 5 prime. And since phosphodiester bond is getting formed from this side, 5 prime to 3 prime side, and the condensation reaction is happening in that way. That's why this is called as the 5 prime to 3 prime. And this tells us that the first nucleotide has a free 5 prime phosphate group, and the last nucleotide has a free 3 hydroxyl phosphate group, and that's why this is called as the 5 prime and 3 prime end. Now, coming to the next part, that is very important also. Why does RNA polymerase reach template strand like 3 prime to 5 prime and synthesize RNA from 5 prime to 3 prime? So as we have seen that this is the nucleotides of the DNA, and in nucleotides once the incoming nucleotide come like NTPs and DNTPs, that contains 3 phosphate group, right? And if you see this NTPs or DNTPs, if this is base and this contains 3 phosphate group, so the phosphate group which is attached with the carbon, first phosphate group which is attached with the carbon, that is called as alpha. Then subsequently, second one will be beta and third one will be gamma. This is the nomenclature in organic chemistry. So what happens here? So 3' OH group is only reactive group. This 3' OH group is only reactive group. So we'll see these things later. And 5' phosphate group is not that much reactive. Okay. So there is a chain reaction that happens over here. You can see that there will be a growing chain or growing strands of the DNA that will take place from 5 prime to 3 prime, because we have seen that the phosphodiester bond is getting formed between the alcoholic group and the phosphate group. So at the 3 prime end, you can see there is a reactive alcoholic group present. Now this alcoholic group will attack on the oxygen of the phosphate group on the gamma carbon. That will, sorry, gamma phosphate group. And once it will attack, what happens? This will create a phosphodiester bond like that here. And then there is a pyrophosphate group that will be getting released. And that pyrophosphate group is when two phosphate group is getting connected with each other. So here you can see there is a phosphodiester bond formation will take place, and the DNA synthesis will happen in that way. The strands of the DNA will start forming like that from 5 prime to 3 prime end, and one pyrophosphate group is getting released. Now, when we talk about this pyrophosphate group, that basically got pyrophosphate enzyme will come and it will convert this into two phosphate group, something like that. And then you can see that the synthesis or nucleotides are added only to the 3 prime end, right? So it will start getting added at the 3 prime end, and that's why the synthesis of this strand takes place from 5 prime end to the 3 prime end, where left side there will be, uh, 5 prime end, and then on the right side there will be a 3 prime end. And this is how the RNA polymerase will start synthesizing the mRNA, something like that. So why do NTPs and DNTPs carry three phosphate group? We have seen in the previous slide there were three phosphate group, alpha, beta, and gamma. And why it is there? Let's try to answer this question. When we talk about NTPs, so in an RNA nucleotide has three parts, that is nitrogenous base, AUGC, ribose sugar, and three phosphate group. So for example, this one is the base, adenine, and you can see there is a triphosphate group, alpha, beta, and gamma. So all of these phosphate groups are highly negatively charged group. You can see three negative charge are there on the oxygen, and both of all of these phosphate group will be repelling each other, right? So when all of this bond breaks, then huge amount of energy will be released because there are a huge repulsion between these phosphate group. And once this energy is getting released, because how incoming nucleotide will get will be getting added with the subsequent sequence of the DNA molecules, and the energy is coming from the release of this phosphate group only. That's why obviously that reaction involves some energy absorption, and that energy is coming from the breaking down of this phosphate group molecules. So I'll stop this lecture here only. If I'll teach something more, it will become so much lengthy. So in today's lecture, to summarize what we have studied that how transcription factor binds with the DNA sequence, a specific sequence of the DNA, and then how it recruits the RNA polymerase. And after recruitment, how nucleotides looks like in the DNA or RNA. And we have also seen that how incoming nucleotide contains three phosphate group, and it basically breaks into pyrophosphate. And finally, the energy released between these repulsion of these phosphate group after breaking down of this bond, that is responsible for adding the subsequent nucleotide to the incoming sequence of the DNA. Okay, so I'll stop this lecture here. See you in the next lecture.